Okay, scratch that. We're going to make this TAC. I want this to be a start codon, and I'll see what that means in a second. So TAC for us would be a start codon. So TA, it's going to be AU, yeah. TAC. I hope you can like visualize this okay. Right, this would be, we don't really care about these because it's the other side, but that would be an A there, that would be a T, that would be a G. TAC, and then I'm just going to make these letters up. Let's go CCG, let's go GAG. Okay, so our gene reads, the template reads TAC, CCG, GAG. You can hear how I'm sort of reading it 3 letters at a time, and that's how genes are actually read in cells. So I'm going to need another color marker, which I forgot to get. I took all these markers from the classroom that last day of school. I'm glad I did. So I'm going to kind of need to either draw over this or erase this a little bit. Definitely do this in pencil, because what's going to happen is RNA, right? RNA, ribonucleic acid, is going to come in here and put corresponding bases, nucleotides. The nucleotide is the little backbone, the phosphate sugar, and then the base is just the nitrogen base, the letter, right? So that would be an A. Now, DNA base pairing, you know by now, right, is A with T, C with G at Cedar Grove, right? RNA base pairing is different. It's the only reason it's different is RNA has no T, it has no thymine. Instead, it has uracil. So it's going to be A with U, C with G. That's one of the major differences between RNA and DNA. So here, RNA cannot put a T because it has no T to give. Gives no T's, you know what I mean? So that's going to be a U. That's going to be a G. I'm getting a little bit off here. That's going to be another G. That's going to be a G. This is going to be a C. This is going to be a C. U. C. Right? What it has just done is it's just popped in these extra nucleotides and synthesized a strand of RNA that we're going to call mRNA. This RNA strand is going to come out here, and here it is out in the cytoplasm. Right? I'm gonna write these letters out again so we can see them clearly. It's AUG, GGC, CUC. Right? That's the code. And these, of course, are these little things sticking up off the strand. I'm not gonna kind of drawing a little sloppy here, but you get the idea. All right, that's how it looks. Right? And it's all held together by this phosphate sugar backbone. Now, this strand here, this is called mRNA, and you're at the M little like that. And that is for messenger RNA. It's taking the genetic message, the message of the gene, out to the ribosome to make the protein. Remember, ribosomes are what makes protein. Messenger RNA. Each 3-letter sequence, AUG, for example, GGC, is a codon. Right? So it's going to come out here, and it's going to, I'm going to move my camera, and we are going to feed this gene right into the ribosome. Here it comes. Now there's 3 binding sites in the ribosome. I'm going to kind of gloss over that detail right now just to, I'll see if I need to make it in a minute. Here comes that AUG, right? And the rest of it's here too, GGC, and CUC. I'm not changing what the strand is. It's just moving through the cytoplasm. Now, out here, there are these little doohickey things that are folds of RNA that are called tRNA. And T stands for transfer RNA. I sort of make them look, I'm going to draw one sort of big down here. I want you to make them look like this. Okay, kind of strange. Now, they're actually made of RNA. So if you were to zoom in on this line, you'd see like the nucleotides were all here, A's, U's, C's, and G's. But there's 3 bases that stick out the bottom. They are exposed like that, right? And now on the other end of the tRNA is an amino acid, right? This is an amino acid, one of 20. So that's how the tRNA is going to look. And then there's tons of these floating around the cytoplasm, right? So when the ribosome it binds with the mRNA, a tRNA is going to float into place with the corresponding or complementary base sequence. So the complementary base sequence, I'm going to bring this tRNA, into the ribosome here. And it's going to be U, A, C. That's what it would be, right? Now, UAC is going to carry the amino acid methionine, right? And what's going to happen here is after that is put its amino acid in place, the tRNA is going to leave the binding site and leave behind the methionine, right? In other words, it's going to disappear and the methionine is left attached to the ribosome. Now the mRNA, I'm going to erase this, is going to move down a slot, right? So we're going to shift this down. I'm just going to make it look like it just shifted. So now that AUG is kind of over here. It's done. The new one coming in, I'm looking at my old strand here, is GGC, GGC, right? CUC is the last codon. Right, this is only 9 letters in this gene. A regular gene would probably be hundreds, sometimes thousands of bases. Now, I don't know off the top of my head what amino acid would be coded for here. I'd have to look at an amino acid decoder, and you're going to learn about that. But I'm going to bring in another tRNA. tRNA is going to be here, right? And it's going to have, you should be able to tell me what's called the anticodon is going to be C, C, G. Now, I'm not exactly sure, again, what I said off the top of my head, what CCG would code for, but like all tRNA molecules, this will be carrying an amino acid. Let's say it's proline, right? Now, that's going to put that next amino acid in place. That tRNA is going to zip on out and leave that behind. So what you're seeing here is a polypeptide. In other words, amino acid chain is being left in place. It will build up, and as more and more amino acids are put in here, this will start poking up out of the ribosome, and then eventually you will get to, at the end of the gene, what's known as a stop codon. It will code for stop, and that polypeptide, that amino acid chain, will be released into the cytoplasm, and you've made your protein.